We presented the ASCEMBL study, which is a randomized trial comparing asciminib, a new kinase inhibitor for CML, compared to the standard of care bosutinib for third-line and later-line patients with chronic myeloid leukemia. Asciminib is a so-called STAMP inhibitor, which means it specifically targets the so-called myristoyl binding site of the ASCEMBL protein. ASCEMBL is autoregulated and inhibits itself by binding the myristoyl end of the protein to a myristoyl binding pocket. And the myristoyl end is lost when ASCEMBL is bound to BCR, forming BCR-ASCEMBL. Therefore, BCR-ASCEMBL is always active. And this inhibition, this auto-inhibition, can be limited by substituting the natural myristoyl the myristoyl tail by a compound, which is asciminib. And this asciminib has been tested in a phase 1 study, published already in the New England Journal last year. And this data was used to design a phase 3 study comparing this asciminib to bosutinib. There was a randomization, which was 2-to-1, 233 patients were included, and the main endpoint of this trial was the major molecular response rate at 24 weeks on treatment. That's very early, acknowledging the fact that the third-line and later-line patients need an immediate response. Otherwise, there's a major risk for acceleration or blast crisis. Therefore, we expect an early response on treatment. The alternatives, of course, are other tyrosine kinase inhibitors or allogeneic